Hello, everybody. My name is Andy Rakeman, and I'm the Vice President of Research at the Lustgarten Foundation, and truly excited to have this very special Lustgarten Live event for you today. Today, we're going to be taking a deeper dive into our understanding of Daraxonrasib, which is a first of its kind RAS-targeted therapy and the first RAS drug to complete a phase 3 trial in pancreatic cancer. Expect a very large audience today. So those of you who are new and this is your first Lustgarten Live, welcome. Thank you for being with us. For those of you who are coming back, welcome back, and glad that you're here to talk with us. And I think you'll see a little bit of a different tone today compared to some of our other Lustgarten Lives, where normally we're talking a lot about research and progress and excitement and hope for the future. Whereas today, we're still going to be talking about research and excitement and progress, but we're also going to start to talk about expectations for the future and what's really going to be different for patients based on this new era of RAS-targeted therapies that we're moving into. So this webinar was really inspired coming off of the exciting phase 3 results that were announced from Revolution Medicines using their drug Daraxonrasib. And then even more excitingly, the announcement from the FDA to grant expanded access for this drug to patients. And then a lot of questions and confusion in the community around, well, what does this all mean? What did the results tell us about the drug? What does expanded access mean? What does it mean to be in a trial versus get drug approval? And what should we be expecting for the future? So that's really where we brought this together today and what we want to talk about. And we hope that you're all going to leave with a much better understanding of, one, what the results were, what did we learn? Importantly, what did the patients experience on the trial, and what did it mean for people's quality of life and experience with the disease? What does it take to move from a compelling trial result to access to patients and use of the drug in the clinic? And what are these different mechanisms that are going on? And also, most importantly, what does this mean for the future of RAS drugs? What other research is ongoing? What does this really going to turn into in the future that's really going to help us to move from hope to expectations to cures for patients moving forward? So this will be a great conversation. It'll be truly inspiring. You're going to hear from some of the leaders that have really been driving this work, both in industry and in academia, and really get a chance to hear from the horse's mouth, so to speak, about what's happening and what's on the cutting edge. But before we jump in, I'll take the privilege of having the camera right now to give you, a little bit of my impressions and why I'm so excited right now. So I had the pleasure of being at the ASCO meeting, the American Association for Clinical Oncologists, when Dr. Brian Wolpin first presented these results to the community. And it was an inspiring moment. As he was going through the data and he showed the slide that had the survival data for patients on this drugs, it was a standing ovation from the room, right? And some of you might imagine what a scientific conference full of researchers and clinicians is normally a fairly staid event, but this was a very emotional response, not just because of the work that represented the tremendous amount of hard work and effort to get that result, but I think really everyone in there looking forward and understanding what this is going to mean for their patients going forward and how this is really hearkening in a new day and a new age for thinking about pancreas cancer treatments. And it's also, really inspiring because we didn't get there just based on hope. We didn't get there based on, wishing. We got there through, hard work and belief that we can do the things that people thought were impossible if we stay dedicated to the science, if we follow the data moving forward. So many of you may have heard KRAS has long been referred to as an undruggable target. And undruggable really just means that the chemists hadn't figured out yet how to make a small molecule or a drug bind to the target that they want to modify with the drug. And that, we hung a lot more on that word yet rather than the word undruggable or impossible. And it was actually work that Lustgarten funded going back to 2014 with an investigator, a chemist at Harvard called Dr. Greg Verdine, that really took that view of saying if we approach this differently, we can get a different result. And it was that hard work and early support of innovative and bold ideas that led to a shift in a new kind of chemistry that enabled drugs like Daraxonrasib and other RAS inhibitors to move forward, getting to that point. So I think when you hear the story of where we are today and what's coming, also think about that, but we're not in a position yet to, sit back and rely on the view of what people are saying is possible right now, that we need to continue to think about how do we challenge those norms and how do we challenge those assumptions about what is possible. And I think you'll hear definitely from the Lustgarten Foundation, that's core in our belief. We're going to follow the data and we're going to continue to take on the hard problems. And I think as you hear from our three guests today, you'll definitely get the impression that these are folks who don't shy away from taking on challenges and hard problems, and we're in good hands of really moving things forward from here. So with that, I'd like to welcome our guests up onto the virtual stage here with me, and I'll give a little introduction before we turn it over and drive the discussion going forward. So it's a great pleasure to be joined here today by, first of all, our moderator, Dr. Elizabeth Jaffe. Dr. Jaffe is not only Lustgarten's chief medical advisor, where she plays a very critical role, and I'm glad to call her a colleague in our efforts to lead research against this disease, but she's also deputy director of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins, co-director of the Skip Raw Pancreatic Cancer Center, associate director of the Bloomberg Kimmel Institute for Cancer Immunotherapy. She's an internationally recognized expert in cancer immunology. She's been the one really talk about taking on hard problems, has led in the belief that immunotherapies for pancreas cancer are going to be a big part of the future and driving that forward. Where she studies, the role of the pancreatic tumor microenvironment in preclinical models and in the clinic to drive forward immunotherapies in patients. And Dr. Jaffe is well known for developing one of the most successful translational pancreatic cancer programs in the country. And that's really been a model for other institutions going forward. We're also joined by Dr. Nilo Azad, who is a professor of oncology and a medical oncologist at Sidney Kimmel Comprehensive Cancer Center at Hopkins. And Dr. Azad's work is really dedicated to translational research and driving clinical trials and investigating new drugs that work both with immunotherapies and vaccines and targeted therapies and small molecules moving forward that are really paving the way for the approaches that we'll take on today. And finally, very pleased to have here Dr. Alan Sandler, who is the Chief Development Officer at Revolution Medicines, the company that's been behind Daraxonrasib. And Dr. Sandler leads the company's global development organization, brings internationally recognized expertise in oncology and drug development and clinical research, and is really doing this building on a distinguished career as both an academic lung cancer oncologist and an industry leader in global drug development. So welcome, Dr. Sandler. Before we turn it over to the team and really get into the conversation here, I want to point out we have a large group and we want to hear from you as well. So there should be a button on the bottom of your screen for Q&A. If you have questions, feel free to put them into the chat and we'll be monitoring those and answering them as allowed and as necessary. But just a reminder, although we do have some very talented physicians here, this isn't the right environment to give advice or answer questions about somebody's personal medical conditions or advice for the future. But very happy to answer questions about the work that we're talking in the future going forward. So with that, I'll turn it over to you, Liz, to lead the discussion. And most of all, really, I was hoping if you could kind of start off and set the stage and tell everyone a little bit about, why this is such an exciting moment for you and kind of what it's meant to see the progress that's being made right now.
Thank you, Andy. Yes. So I've been working in this disease, as everyone on the panel knows, for over 30 years. And I got started because my father's brother in 1986 developed pancreatic cancer. I was just training to become a doctor then. I didn't know a lot about cancer. Obviously, I'm an oncologist now. But one thing I did know was that it was a death sentence. And I just remember my dad's call and we cried on the phone. And sure enough, my uncle passed within 3 months, not without experiencing pain, but definitely without any hope and without any options. And it's taken over 30 years to get to this point. And now it's an unprecedented time in the history of pancreatic cancer because for the first time we have a drug that actually works in this patient population. And you're going to hear from the experts today just how well this drug is working, but also we're just at the beginning. This is really the start of a new era. And so what you're going to hear today is really how do we go from this hope, from this really important finding, this important drug, to really making a big difference, as Andy said, from hope to being able to take this cancer from a death sentence to a chronic disease, and hopefully in the future to cures. So with that, I'm going to start, and actually I'm going to start with Alan. And Alan, can you tell everyone what the results of this clinical trial means for patients?
Sure. Thanks, Liz. And again, thanks for having me on this esteemed panel. And it's really, it's a pleasure to be able to talk about this and to have some positive data for pancreatic cancer patients. I think you've all sort of begun and highlighted what it means. This is something that there just really weren't many treatments for patients with metastatic pancreatic cancer, and outside of modest benefit from chemotherapy with associated toxicities. So I think that some of the take-home messages from this, of course, number one is the fact that the survival when compared to standard of care in these, and these are previously treated pancreatic cancer patients, so they had had first-line chemotherapy. So expectations in this setting are roughly 6-7 months. And so this was a randomized study comparing Daraxonrasib, which is an oral agent. It's given daily, and compared to chemotherapy. And so the results showed a doubling of the median survival. But if you look at something a little fancier with statistics, the hazard ratio was 0.4, so a 60% reduction overall in the likelihood of dying with the use of Daraxonrasib in this setting. So first and foremost, overall survival is critically important. But two other things I'd like to comment upon is one, that the safety profile, of course, was acceptable. It was and well tolerated by patients, and certainly different from that of chemotherapy. And I know Nilo can talk about this in more detail, having had more experience individually with patients. But the third one, I think that's very important, and is that of patient-reported outcomes, or quality, also known as quality of life. And we also showed that data there, that there was a similar improvement in two measurables. One was the time to deterioration in pain, and the other was the time to deterioration in global quality of life. Both of those were impacted favorably by Daraxonrasib. And although it seems logical to assume that they would go hand in hand, as we all know on the panel, that doesn't, and many in the audience, I'm sure as well, that doesn't always happen. So not only are patients living longer, but they're feeling better longer in this setting. So I think that, and then of course, lastly, hope is the other prevailing thought for patients. So I'll stop talking, let others contribute.
Well,
Thank you.
One of the things that I think is really important to mention about the magnitude of the benefit that we saw in this trial, Alan, you mentioned that this was in patients who had already been treated, but we have over 20 years been trying, and longer, been trying to figure out what can we do for patients with the treatments that we have, and that's really been standardly multi-agent chemotherapy that's pretty toxic, as many people in the audience know, and we definitely see. And so when you give patients treatment, the earlier that you give them treatment, this is kind of a truism across cancers. So the earlier that you treat patients, the better that patients do with the same treatment. And so Daraxonrasib, in this highly pre-treated setting where patients had already been exposed to chemotherapy, the toxicity, most of them they already had tumor growth, it did better than when you give chemotherapy to patients who've never had treatment before. So it actually did better even in later lines of therapy and I know we're going to be talking more about the future, but that's so exciting because when you then think about how we can move these drugs into having more benefit, both by themselves and in combination, moving them earlier, right when patients first get a diagnosis, I think there's real hope that we're going to do even better than what we have seen with the Daraxonrasib Resolute 302 trial.
Yeah, and I would just add real briefly, just to put a number on that. I mentioned the doubling of survival was about 13.2 months for the median, and this actually was the first time, again, as you mentioned, that we'd ever exceeded 1 year in any, even including first-line pancreatic cancer. So that impact that you were describing, that just puts a little bit of a quantification on it, as you mentioned. But yes, thank you.
Vanilla, can you qualify too? I think there's some confusion as to who currently can have access to this drug. And we could talk about the extended access. So maybe Alan could talk about the extended access, and then you could mention who will get access to this drug currently.
Sure. Alan, why don't you start?
Alan.
Okay, sure. So the extended access program, it's also been called compassionate use, but this is something that is, and it's great to have an opportunity to clarify, because everyone virtually everybody with metastatic pancreatic cancer, previously treated metastatic pancreatic cancer, has access to this. It is driven, however, by the physician, the patient and the physician. We have on our website an opportunity to go in and get the ball rolling, get it started, led by our group, our medical affairs, our global head, Mary Pinderschenk. But we basically then it is initiated by the patient and physician. And then, of course, there are some administrative aspects that have to be accomplished at the site itself, going through the IRB, because it's sort of a light touch clinical trial. It's not a clinical trial, but there are some administrative aspects we do follow, looking for safety issues along the way, just to keep tabs of that aspect minimal with that. But we're happy to report that the process is working, and drug is actually now being shipped to various sites as well. So, and of course, there has been a very good outpouring of interest in this. And I'll pause here, see if there's other questions. And Neil, please feel free to.
Vanilla, just if you could explain what patient population will have.
Yeah. So because Daraxonrasib in RASolute 302 was tested and has shown this benefit in people with pancreatic cancer who have already been exposed to chemotherapy in the metastatic setting, that is who the early access program is for. So it is not, I see a lot of questions in the chat right now about what if you've got, resected disease, what if you're in surveillance and you're NED? Thank God you're in NED, but this expanded access is not for that group of patients. It's for patients with advanced disease that have already had chemotherapy or a first-line treatment at least, but you can have more than just one line of therapy as well. And so, that has, I do want to say, Revolution Medicine has done so much to try to get access for patients as quickly as possible. Even the fact that the early access application was filed to the FDA so quickly as soon as the results were reported, and then the FDA, unheard of, in 2 days said, yes, this is important. The data is so exciting. That said, the need and the desire for this drug right now is so huge. And I would not be surprised if there are people in this audience that are trying to figure out how is it that we can get access, where are the sites? And we are all learning in real time. We're all doing our best. Liz knows that here at Johns Hopkins, our internal list has over 50 patients on it, and it's continuing to grow. And so I think working with your doctor to really figure out the steps that go forward is really the best way to get access to this agent. And we also hope that timeline of how long this EAP is going to be in place before the drug is approved, fingers crossed that's going to be quickly. But there is a lot more interest and excitement and need for the drug right now. And the process is coming through, but it hasn't, it was a little surprising how quickly it happened. And so we're all dealing with that and trying our best.
Right. And the extended access is an in-between, or it's to stabilize and be able to provide the drug until this drug gets FDA approval. So Alan, when do you expect that? I know you can't give a date, but is it this year? Is it early next year? When do you think we'll see that?
Yeah. So I think, what I can comment upon is that, this is, we also have the voucher that is also a means for the FDA to try and accelerate the process. We're actively working with them. It's what's called a rolling submission. So we're taking, completing sections, sending it to them so they can begin the process without waiting for the entire package to be there. I can't really comment on when it's going to be, as I mentioned, but this should be sooner than the traditional approval process. And we're working both sides very, very collaboratively with the FDA. The FDA has been very responsive, and we're working to get this out as quickly as we certainly can.
And I do want to just point out that actually the Lustgarten Foundation helped to get that voucher for this drug. We
Ah,
saw early on that this was going to be needed, and I give the Lustgarten Foundation a lot of credit for trying to support you as well.
yes. Thank you.
Before we move on to next steps and where things are going, Neil, can you just talk a little bit about what do patients experience? How is this drug delivered? How do you monitor the drug? What should they be expecting?
Yeah, so Daraxonrasib is an oral agent, and so it's pills that you take once a day. And so patients who have gone on the study, there have been so many wonderful moments of being able to treat patients with these drugs. First of all, not only did the data show that there was a doubling of overall survival, but if you look at overall how patients did, 90% or more of patients benefited from the drug. Their disease was under control. And what we saw as doctors that were treating patients was that their symptoms improved very quickly. So we had patients who came in who had pain or who had fluid buildup, and within weeks we were seeing, sometimes days, improvement in those symptoms. The drug itself does cause side effects though, right? So, and I think in terms of this is what was reported, and it is absolutely what we saw, is that the most common side effect that we saw with Daraxonrasib is a rash. We, as investigators in Revolution Medicine, has been working for years as this drug has been developed to find, the right way that we could prophylax and really help mitigate that rash. And so before patients even get started on Daraxonrasib, we give them medications, topical and oral, to try to keep that rash down at a minimum. And then once you're on treatment, a patient can have a mild rash, they can have a severe rash. More commonly, it's mild or moderate. The severe rash is not as common. We have had some publicity around a couple of folks that have, given interviews and we've seen some very extreme rashes. That's less common, but always able to be mitigated by holding and dose reducing. And so there's a lot that we can do, but the rash is the most common side effect. Some oral ulcers as well is something that we see that we sometimes have to hold or do dose reductions for. But generally speaking, it's a pretty good drug in terms of tolerability. The quality of life data supports that as well. And another thing to just always remember is that if people are able to be on a drug for many, many months and it provides, on average, many, many months of benefit, that's telling you that it's tolerable enough for people to do that, right? So the data shows you how tolerable it is, because if it were not tolerable, people would not be able to stay on for as long as they are. So a lot of excitement around being able to make people feel better, even with the side effects.
Right. And we know that drugs that work well in advanced disease and chemotherapy resistance disease is going to work better at earlier stages. So maybe it's time to start talking about what is Revolution Medicine's doing currently to test this drug in earlier stage diseases, Alan.
Sure. So there's an ongoing study, alluding to going back to Nilo's view that the earlier we're able to treat these patients, the more of an impact we're going to be able to have. So there's a first-line study, which is a 3-arm study, where we're actually looking at Daraxonrasib as a single agent or in combination with chemotherapy versus chemotherapy. So it'll address 2 issues, the role of monotherapy and the success of that, and then does the addition of chemotherapy provide an additional advantage. Another point to look at is in the adjuvant setting. And as has been mentioned, there were some questions in the audience, of course, about the EAP for this. And although the EAP is not available for this group of patients, there's a clinical trial that we have that's ongoing and accruing patients for those who have had a resection, a complete resection, and have had some form of perioperative therapy. And then they're randomized to 2 years of Daraxonrasib versus no further treatment. So another means where, in this particular case, we actually, in addition to extending life, we'd love to be able to see that we might be able to cure more patients along the way in this particular setting.
And I just wanted to mention that the reason that these trials were designed, especially the first-line study, was because there were cohorts of patients that we enrolled in the original trials that gave Daraxonrasib alone to patients who were just newly diagnosed, and also gave Daraxonrasib in combination with gemcitabine and Abraxane. And those data have been reported. Now it's small numbers of patients, right? So we've got a few dozen patients versus a trial like Resolute 302 that's hundreds of patients. But in that small number of patients, we saw really exciting results that really made us feel that this first line approach may be the better approach. And so this isn't just theoretical that we think it would be better in the first line, but there is data to support that response rates go up and that more people seem to be benefiting in the first line. And now we just need to do it in a bigger group of patients to make sure that those results are real.
And is the combination with chemotherapy still tolerable, or are there other side effects?
Yes, I mean, when you combine Daraxonrasib with gemcitabine and Abraxane, which is how it's being combined, you get the additional side effects of gemcitabine and Abraxane. But this was tested in a phase 1 trial to find the right dose of how to be able to give this in a tolerable way. We were able to do that, and we've now, we as a community have been able to deploy a multi-hundred patient trial that's going to be able to test it. So you do get the added side effects, but we hope that that's going to end up being with improved benefit. And after a few months, you can then go on to Daraxonrasib alone as a maintenance strategy. So it's kind of the idea of let's go at it hard up front, that arm. So let's go at it hard up front with both and then pull away to just the single agent alone versus do you just give the single agent alone and that is as good? Now we'll know which way is better.
Yeah. It almost looks at a sequential versus concurrent approach, because undoubtedly those who have Daraxonrasib as monotherapy will go on to get chemotherapy upon progression off study. So there's a somewhat of a secondary look there.
I wanted to follow up again, Alan, on your comment about in the no evidence of disease, it's a randomized trial to either nothing or the drug. And for those who are in that situation, of course, they want the drug. I realize this is how you get a drug approved, but can you just talk about how will these patients have access to this drug in the future if they get on the control arm? Can you just mention a little bit about that?
Sure. So one thing to note and just emphasize is these patients will have had the opportunity to have perioperative chemotherapy, either before, after, or both, which is current standard of care. So everybody will have received standard of care. And I think this is important for any patient who might be thinking of joining this particular study. And therefore, then they have about a 50% chance of getting Daraxonrasib in this setting. And as of now, that would be the only way that one would have that opportunity. To answer your question, would they be able to receive it subsequently? Yes, if, depending upon what our label looks like, how our first line study reads out, they'll be able to receive it either perhaps in the second line setting, ultimately perhaps in the front line setting, depending upon where they are in the trial and where we are in the process of our studies and discussions with the FDA.
And I do want to mention that I completely understand the desire to want to get Daraxonrasib after you have surgery and have had your chemotherapy. And I think that it's very understandable, and I'm very hopeful that Daraxonrasib will add something to patients. We only want to keep improving outcomes. But it is important to note that there are many drugs that have worked in the metastatic setting in various cancer types that when you move them into the adjuvant setting, they don't necessarily provide that benefit. And so that's why it's so important to do the test. We can't just make the assumption that because something works in one setting, that it's going to work in a very different setting. And so I just think that's important to think about, especially when you go into any randomized trial.
Yes, absolutely. And toxicities are real. So, and there's always going to be severe toxicities in some patients. So can you, Nilo, talk a little bit more about what other drugs are starting to be developed down the pathway to approval that target this pathway?
Yeah, so KRAS mutations are not all the same. And so we talk about mutant KRAS or mutated KRAS, but actually you can have many different mutations in KRAS that result in the same problem, that KRAS is turned on all the time, and so the cancer cell survives. Daraxonrasib is a drug that targets many multiple KRAS mutations. So if no matter what KRAS mutation you have, you can take Daraxonrasib and it hopefully is going to provide benefit. But that said, we know that pancreatic cancer subdivides into different kinds of KRAS mutations. So the most common KRAS mutation is a KRAS G12D mutation. That's just the exact location in the DNA where the mutation occurs. About 40% of patients have that mutation with pancreatic cancer. So now there are drugs, many drugs that are being developed that target that particular mutation. For example, Zoldonrasib, which is Revolution Medicine's drug, is a drug that targets that very particular KRAS mutation. If you have one of the others, it's not going to work for you. But if you have KRAS G12D, beautiful data that's been reported showing that this drug is really active and less toxic because it's only targeting that one mutation. And so there's presently an ongoing phase 3 trial in the first line with Zoldonrasib. So if a patient has a KRAS G12D mutation, they can take Zoldonrasib with chemotherapy versus chemotherapy alone. So that's the Resolute 305 trial, really exciting study. I know Alan's here from RevMed, but let me just say many other KRAS agents are out there as well. There are some drugs that target multiple KRAS mutations. Data is just starting to emerge. You know, the other drug that has targets multiple KRAS where we have data that's been reported has not really been reported. There's a press release that says we've got similar activity to Daraxonrasib. So we're waiting to see that data for the academic community really be able to look and see, like, is it the same? What's the toxicity? Multiple G12D inhibitors from multiple companies. RevMed has a G12V inhibitor. So there's many different drugs from many companies, over 30 or 40 drugs on the last time that I put up a slide in one of my talks that are being developed. And then there's also drugs that are targeting KRAS in different ways. You know, Liz and I work with KRAS vaccines. So we'll talk about that a little bit more, I'm sure, in a bit, because we're excited about combining those. But, you know, there's ways to hit KRAS beyond just inhibiting KRAS, but when we can try to use KRAS as a signal to the immune system as well. So lots of exciting work in this space, and a lot of it is being tested in different lines of therapy for patients with pancreatic cancer.
So this is a really good segue into drug resistance. So we're really excited by the doubling in life expectancy in the second line, but the reality is eventually the majority of patients will recur. Can you talk a little bit of what we're learning about the mechanisms of resistance and how are we going to combat those mechanisms?
So the bulk of the data that we have about resistance in patients that have been treated with a KRAS inhibitor actually comes from a more rare KRAS mutation called KRAS G12C. It's only present in 1% to 2% of pancreatic cancer, but it's more common in lung cancer and colon cancer. And those were the first drugs that really came into the clinic and were approved. And there's been very beautiful, elegant data that has looked at sequencing patients' tumors and their blood after their cancer grows on a KRAS G12C inhibitor and has seen different mutations that emerge that predict resistance. That you would say, okay, a different mutation in a different KRAS mutation, or an amplification, an increase in the path in a different pathway, which is actually even more common. And so now we're doing the same thing with Daraxonrasib and with KRAS inhibitors. And in fact, you know, the Lustgarten Foundation has paired with Revolution Medicine and with the Breakthrough Cancer Foundation. So this is a kind of a trifecta, I feel like, of industry, academic, and philanthropy that has come together. And really, we've over the last couple of years enrolled an additional cohort of patients where we are profiling patients at resistance to find in pancreas cancer, what are the mechanisms of resistance? And so then how can we work on combinations that could overcome that?
Great. Thank you. And did you want to add anything to that, Alan? I know the company has been working also on resistance mechanisms.
Yeah, I mean, I think Neil covered it very well. I was going to emphasize the amplification is one dominant way, but the other mutations that develop. And I think the key overriding theme appears to be that once RAS is important, that the resistance, even with resistance, RAS remains the key driver. And I think that's sort of a key element in terms of the approach. And so other things to be thinking about is and what we're thinking is perhaps continuing in some fashion with the RAS, targeting RAS, and then adding to it, as opposed to what is often done in other settings in oncology, where you try something in first line progression, you stop and move to something completely different. I think keeping the trying to keep your foot on the brake for RAS, it may well turn to be critically important. But studies will be necessary to prove or disprove that.
Can either of you mention any potential combinations that you think will be the earliest ones tested? You want to start, Nilo?
Yeah, I was like, I sure can. And so, I think as we profile what happens at the time of resistance, I think we're going to see rational combinations that put these drugs together with the areas of resistance. So if you get an amplification of a different circuit, that might be the combination that you consider in certain patients, and that data is developing. I also think that there is an important sequencing question that does speak to combination therapies as well. And so, for example, when you take a drug like zoldonrasib, which hits G12D, is it better to give that drug in combination, or is it better to give that drug sequentially, give zoldonrasib first and then come in with daraxonrasib at the time of resistance? So those are combinations that are, I think, very important. Just in the last couple of weeks, amazing data coming out of Tango Therapeutics, looking at combining a PRMT5 inhibitor in combination with Revolution Medicines agent. This is because PRMT5 inhibitors target patients that have something called MTAP deletion. That's about 20% of patients. Those patients also are KRAS mutated. So there is a hope that if you put those two together, that was going to be better than either alone. I think we were all shocked and excited by how much benefit it looked. It looked like it was really maybe even synergistic, not just two things adding 1 + 1 equaling 2, but 1 + 1 equaling 3. And so, combinations where we look at other abnormal circuits and combine them together. BRCA mutated patients, should we be putting PARP inhibitors together with a KRAS inhibitor, for example? And I really hope we're going to have a whole segment on the immunotherapy piece. So I'm not going to even answer that yet, because we're going to talk about that coming up too.
Yes, I would add one other aspect. I think that was very well said. Is we have an agent RMO55, which again is something that works on the amplification side. It's a catalytic agent that accelerates the catalytic event from GTP to GDP, basically going from on to the off state, which may well serve to overcome, and certainly in the preclinical state serves to overcome some of that resistance that we've talked about. So that's something that's not yet in the clinic, but looking forward to that.
That sounds great. So Nilo, I'm going to now ask you, so what does the inhibitor of KRAS mutations do to the tumors themselves to make us think that it would be good to combine with immunotherapy? And how are we thinking about that?
So concurrently to the development of these agents, there were multiple very astute laboratory investigators, including yourself, that were really interested in saying, look, if we are going to dampen a circuit that we know is very important to drive this cancer, what is that going to do to the area around the tumor? Because we knew from before that cancers that were KRAS mutated seemed to have a tumor microenvironment in terms of the area right around the tumor that didn't have the kinds of immune cells that you would want for an anti-cancer response. So you had immune cells that were really more negative, and you didn't have the cells that you really want. And so in that setting, what does KRAS inhibition do? And so over the last few years, what we've seen is multiple groups that really focus on this story, seeing that when you take tumors in the laboratory, right? So in mice or in animal models, but when you do that, what you see is actually a change in that tumor microenvironment where now immune cells start flocking to that area, immune cells that you want, the ones that are anti-tumor immune cells, and the bad immune cells, the ones that are always dampening the immune response, those seem to be changing. They become less functional. And the cancer cell itself seems to be able to flag the immune system better. And so those were the first results. There was a group out of University of Pennsylvania, Ben Stanger's group, that was also able to demonstrate that if you took an animal and you took out its immune system, now the KRAS inhibitor didn't work as well. So that really suggested that you need the immune system to have the best response as well. And then multiple groups started saying, well, what if we combine these drugs in animals with immune checkpoints? What if we combine them with vaccines? And so that data now has been percolating over the last year and really shown pretty dramatic results in the laboratory about when you combine things with immune modulating agents. And one last thing, the reason this is so important is because, as you mentioned, Liz, patients become resistant to these therapies over time. The hope is that if you activate nature's best anti-cancer system that is with us from the time that we're born and keeps us from developing cancer so many times until it doesn't, if we can activate that, then we can get a durable response that could last forever. And for patients that have been treated with immunotherapy successfully in all different cancers, immunotherapy has been able to take cancers that were thought were incurable and turn them into chronic diseases, sometimes even cure them. That's what we want.
Right. And just to add, I mean, this beautiful way of explaining it, the immune system has the ability to not only provide memory, so you get rid of the cancer. If it starts to come back, the immune system has the ability to also ramp up and deal with what's coming back. So it's really powerful and could very well be of benefit to patients who receive these drugs. There's a lot of discussion in the chat, Alan, about the concern over will there be enough supply of the drug for extended access, and how are you thinking about that? How should we, be thinking about it locally, and then also can international patients get access to it?
Yeah, I'll start with the supply. We've certainly have anticipated high interest, and we've definitely set aside a drug for the EAP, and we will ensure that those patients that are accepted into the EAP are able to receive treatment. I think the other part of the question was outside the U.S. The EAP is a U.S.-based program because, again, as you mentioned earlier in the talk, it's designed for those drugs, those rare drugs that look so good early on in completing a phase 3 study that it bridges the time until the drug is actually approved. So that's a U.S.-based program now. There are those types of programs outside the U.S., and those are things that we will be working on, but those are country-based, so you have to work with each individual country. And you do not have to be a U.S. citizen to receive the drug, but you do have to be treated by a U.S.-based physician in order to receive treatment. So I would also add for those patients outside the U.S., and a critically important point is that we have a number of studies, as we've talked about, that are ongoing, and we have greatly enlarged our global footprint. So many sites throughout the world outside of the U.S. have access to multiple clinical trials that include Daraxonrasib, include some of our other agents like Zoldonrasib as well. And so, I would urge patients to pay attention to that and please try and look into the opportunity for clinical trial participation. And again, we'll be working with the various agencies outside of the U.S. Moving forward on plans for compassionate use out that way.
So with a drug like this, and I know you can't say for sure, but after FDA approval, I mean, how fast you think? Is it a year? Is it 6 months for EU approval or European country approval or Canadian approval? Does that happen pretty quickly with an exceptional drug like this drug?
Yeah, so unfortunately, of course, I can't give you the precise answers that everybody would like, but what I can say is Other countries, I think, have viewed the data that they've seen at ASCO in a similar light to the US. So I think that they're as excited and enthusiastic to do that. There is a standard process that one has to go through. Most of the Europeans wait, dependent upon the US approval, and then the process begins moving forward. And we're working as quickly as possible with the various countries to get this drug Daraxonrasib out to patients with pancreatic cancer. We're working hard on that and feel what the patients are feeling as well, and we're working hard, and I understand again that the governments that we're working with feel that same way.
Okay, and there's a lot of questions about the specifics of patients who can receive the drug currently. Do they have to have had surgery before? Does it work in the liver? Nilo, do you want to answer these questions?
They do. Nobody has to have had surgery before. They just, the eligibility is actually very nice in terms of being inclusive. So patients just need to have had some exposure to chemotherapy before because of having metastatic disease. What was the other question? I'm so sorry, Liz.
Does it work in the liver?
Oh yeah. So this agent, we've seen activity in all the organs that people can have metastatic deposits in: lungs, liver, abdominal cavity. That's been pretty amazing. Peritoneal disease has been kind of a hard thing for us to treat with chemotherapy. Has not worked as well, and we've seen really nice responses. So no matter where the metastatic deposits are, we feel really good about this drug.
Great. And then some of the other questions, has it been tested with chemotherapy other than Gemabraxane, for instance, FOLFIRINOX? And if yes or if no, why?
So, do you want to take it?
I think we have done studies with other chemotherapy agents. We felt that the best tolerated and one that had significant activity was with the gemcitabine and nab-paclitaxel. So I think Nilo, did you want to add?
Yeah, I mean, we as an investigator on the studies, all the investigators sat with Revolution Medicine and looked over the data every time that we enrolled patients on the different arms to test these different combinations. We all agreed that this was the right combination to move forward with.
Great. And then there's a question about immunocompromised individuals. So what are the current guidelines for being accepted into the clinical trials relevant to immunocompromised states?
I mean, I consider cancer patients to be immunocompromised. We know the immune system of cancer patients, especially those who've already had chemotherapy, is immunocompromised. So I'm not sure if patients mean something.
Well, I think when you mentioned that in mouse studies, when you give a mouse the drug and they don't have immune cells.
Yeah. This was, those were studies in animals where you basically knock out the total immune system of the animal. You're not a little compromised. The animal just doesn't even have an immune system at all. And that just showed that having an immune system helps these drugs work better. But all of our patients that were on Resolute 302 had prior exposure to chemotherapy with the immunocompromised status that kind of comes with a little bit of having that kind of chemotherapy. Look how well they did, right? So
yeah, the only thing I would add to that is, of course, there are certain parameters that have to be met to go on a clinical trial, adequate white cells, and there may be some diseases, that concurrent illnesses that might render you ineligible, but those are generally rare. And I would urge, without going to individual patients, but urge you to talk to your physician to see whether or not you'd be able to qualify.
And then another question about the extended access. So is it only going to be available at NCI designated centers, or is it available by community oncology groups?
Yeah, that's a great question. I wished I would have mentioned it specifically, actually, myself. No, it is everywhere. All that you have to have is a certified physician that sets forward to the application, and it could be community-based or NCI-based. And we have sites from both venues that are open and activated already.
Great. And then another, I think, good question here. And by the way, the audience is asking wonderful questions. But so what is Revolution Medicines doing next to improve on the drug, make it more safe? Can you talk a little bit in just general terms where you see drug development going in this pathway?
Yes, so we have that tri-complex platform that we've discussed, and so we like to think of this, and I know Mark Goldberg, our CEO, loves to use the baseball analogy that we're maybe in the second or third inning, so we're getting started. There's been some success, and we're continuing to build upon it. We have the mutant selective agents that Nilos mentioned. We also have the sort of second and third generation as our chemists and scientists continue to look to see what we can do differently. We have the different approach, the catalytic inhibitor. So yes, it's a great question. We're going to maximize what we can do with agents such as Daraxonrasib, but we're also looking to build upon that as well with this platform, looking at either whether it be other targets, looking at how we improve the ones that we have already, and looking also preclinical combinations, what kind of combinations make sense, and that type of thing as well.
That sounds great. So I want to thank you both for answering all my questions. I'm going to turn it over to Andy to finish up this session.
Thank you.
Thank you, Liz, Nilo, Alan. That was a wonderful discussion. And thank you to the community for, as Liz pointed out, so many questions, so many wonderful questions. I think we've been trying to type in answers as furiously as we can. Liz did a great job of trying to bring some of them forward. I think it clearly shows the excitement that we have and the passion that the three of you have in driving this forward, I think also comes through very clearly. So thank you so much for joining us. I think we touched on hopefully a lot of those issues that came from the beginning. People have a little bit clearer understanding about what it took to get to this data where we're at a place and moving forwards. I know that there's still a lot of questions from folks about, well, how do I do it practically? How do I get access? When? And Alan, I know that's always the hardest question. What day is this going to land? And we are getting there. But I think what hopefully people understand is that even with some success, nobody on this webinar is slowing down. We're pushing forward. We've seen fantastic results in these advanced patients with a very hard to treat disease, but we're also looking at, going to earlier stages and earlier treatment lines. And the data is coming on those, and we'll continue to learn more. We know that resistance and combinations are going to be an issue, and we're driving towards that. I'm glad we came to the discussion about immunotherapy combinations because of the group here on the call. That certainly will be a future for this and one that we'll be looking at. And I hope everyone sees, that this is not a group and this is not a community that backs down from the hard questions and the hard problems, because we know our patients are facing a much harder disease and that we need to be, as brave or braver than they have been in driving this forward. So Dr. Azad, Dr. Jaffe, Dr. Sandler, thank you so much for being with us, taking the time to explain this and convey your excitement and your passion to our community. We really appreciate you joining us for this today. Thank you so much.
Thanks for having me.
Thanks so much.
And again, lastly, to close out, thank you, even with as much thanks to the community for showing up to hear about this, to come in with the excellent questions, and for continuing to be a part of this group that is really driving us to bring to a point, as Liz pointed out so well in the beginning, to take pancreas cancer from a death sentence to a manageable disease to a curable disease. And I think, we're not there yet, but we're well on the way, and we're not growing tired, and we're going to continue to push forward and do more and more about that. We hope that you stay engaged with us to continue to follow along with these webinars. We are planning again our next LustgartenLIVE in September 16th, where we'll be talking about how breakthroughs often come from the most unexpected people and places, right? And I think this is the story we talked about today is a great example of how we get to these breakthroughs because people came in and took a risk. They took on a hard problem, and then we had the fortitude to see it through to an end, that it becomes available to patients. And we know that this is not the last issue that we're going to need to do that with. So you'll hear from more about how the foundation and how our researchers are looking for what are those next big hurdles down the road that we're taking on. So we hope to see you then. And until then, I hope everyone has a wonderful summer. And thanks again for all of your support and taking the time to spend it with us this afternoon.